Hey there, science enthusiasts and curious minds. Welcome to this YouTube channel, and we will be diving deep into the fascinating world of cell biology. Today, we have an exciting topic to explore, which will be the structure, function, and membrane trafficking of the cytoplasmic membranes. Have you ever wondered how our cells function, communicate, and transport essential materials within? Well, you're in the right place. In this video, we will take a journey into the intricate world of the cytoplasmic membranes where the action truly happens. The title for today's lecture series will be Bridging Boundaries, the Structure, Function, and Membrane Trafficking of the Cytoplasmic Membranes. This is Sir Stephen C. Capilitan of the Biology Department, and we will discuss the fascinating world of the cytoplasmic membranes. Before the start of the 20th century, stained animal tissue sections hinted at an extensive membrane network in the cytoplasm. And in history, in 1940s specifically, electron microscope revealed a diverse array of membrane-bound structures in the cytoplasm of the most eukaryotes. And with this, they saw long channels bounded by membranes that radiate through the cytoplasm to forming an interconnected networks of canals, and they saw these stacks of flattened membrane-bound sacs. These studies and subsequent biochemical studies show that the eukaryotic cell cytoplasm was subdivided into a variety of distinct membrane-bound compartments, and these investigators found different organelles in diverse cells, from yeast to multicellular plants and animals, and each of these organelles contains a very particular complement of proteins and is very specialized for particular types of activities. Remember that the cytoplasm of a cell is divided into a specialized membranous compartments where different activities can take place independent of one another. These may view as a stable structures, but in fact they are dynamic compartments that are in continual flux. Organelles studied will be the endoplasmic reticulum, we have the Golgi complex, endosomes, lysosomes, vacuoles. Taken together, these organelles form the endomembrane system and function as a part of a coordinated unit. So let's take a look at the overview of your endomembrane system. We have here the figure on your right will be the membrane-bound compartments of the cytoplasm. As what I've said earlier, these endomembrane systems act as a dynamic or coordinated unit. They are distinct compartments bounded by those membrane barriers and contain specialized proteins for a lot of particular activities. Showing you a figure on your right will be the overview of your biosynthetic and secretory pathways that unite the endomembranes into a dynamic interconnected network. You will see that there are materials here that is being packaged in small membrane-bounded transport vesicles, which will coming from the bud from a donor membrane compartment and via the motor proteins on microtubules and microfilaments of the cytoskeleton. They are helping it to transport into the acceptor compartment. And when the acceptor compartment has fused the membrane of these motor proteins and vesicles, that's the reason why it is called a dynamic interconnected network. There are also pathways and secretion modes. The first one is the biosynthetic pathway, where proteins are synthesized in the endoplasmic reticulum. They have been modified at the Golgi complex and they are being transported to various destinations. Second is a secretory pathway. Proteins are created in the ER and discharged from the cell. We have the endocytic pathway. Materials move from the outer surface of the cell to compartments such as your endosomes and lysosomes. We will be discussing and talking about more of these materials and organelles later on in this section. We could also have constitutive secretion, where materials are transported in secretory vesicles and discharged in a continual manner. We have regulated secretion, where materials are being stored in vesicles and discharged when in response to a stimulus. So this is more about like in connection with your nervous system. This is an example of an overview of a biosynthetic or secretory pathways that unite the endomembranes into a dynamic interconnected network. So you will see here different organelles, membrane-bound organelles such as your nucleus. We have the rough endoplasmic reticulum. We have your Golgi complex, and it has different phases: the trans-Golgi network, the trans-cisterna, the medial cisterna, and the cis cisternum. And we have your secretory granules found along the way, and the processes of exocytosis and endocytosis, the regulated secretory pathway, your constitutive secretory pathway, and your endocytic pathways. So these pathways and secretion modes, for example, in regulated secretion, happens in your endocrine cells where they release hormones, such as your pancreatic acinar cells. These are also digestive enzymes, and even in the nerve cells when they transmit these molecules called the neurotransmitters. Secreted materials can be stored in large, densely packed, membrane-bound secretory granules. We have also proteins, lipids, and a lot of complex sugars that are transported through the cell along the biosynthetic or your secretory pathway. There are a lot of types of cargo that are being routed to their appropriate cellular destinations by sorting signals encoded in the amino acid sequence of the proteins or in the attached oligosaccharide. So there are sorting signal sequences which are usually found in the N terminal and the C terminal portion of your amino acids. So there are a few approaches to study the endomembranes, and the first one is your autoradiography, where it visualizes your biochemical processes by radioactively labeling your molecules. We have Palad and Jameson that has incorporated these radio-labeled amino acids into pancreatic enzymes, and they were able to localize some of its cellular proteins. This showed that the ER as the site of secretory protein synthesis happening. And they have also performed a pulse chase experiment to determine the intracellular path of secretory proteins from their site of synthesis going out to their site of discharge. So you can see here the autoradiography, which reveals the sites of synthesis and subsequent transport of your secretory proteins. You have here, they have tagged silver grains, and then these are being viewed under the autoradiography. The next few approach to study the endomembrane is with the use of the fluorescent proteins. We call it GFP tagging, which allows the microscope viewing of the protein movement in living cells. It means green fluorescent protein. So cells infected with a strain of vesicular stomatitis virus, which has a viral GFP gene fusion. So these viral genes will take over the machinery of the cell. So remember that they are intracellular obligate parasites. So they are infecting, or they are hijacking the cellular mechanisms of your cell, and the production of and the movement of these viral proteins will be monitored via the fluorescent GFPs. An example of this in this figure, your viral genome, or your VSV, contains temperature-sensitive VSVG gene, which is fused to the green fluorescent protein gene. So you will see the movement, okay? As you decrease the temperature, so we start with 40 degrees Celsius and then 32 degrees Celsius, and you will see that these proteins, the green proteins, are being viewed in this particular portion where it started off with the rough endoplasmic reticulum, and it went to the surface or the membranes of your Golgi apparatus. Next will be the insights from the analysis of your subcellular fractions, where they've used your cell homogenization fragments. So the cell homogenization fragments will reveal this cytoplasmic membrane. So we have vesicles that derive from the endomembrane system, form similar sized vesicles referred to as the microsomes. And the microsomal fraction can be fractionated into smooth and rough membrane fractions. Once they have isolated, the biochemical composition of different lipid and protein fractions can be determined. So you will see here the homogenate and the whole cells. And then you will see the post-nuclear supernatant will float on top. We will centrifuge them again to for 20 minutes, and the whole cells and the bigger chunks of cells and organelles will stay at the bottom. We will transfer the post-nuclear supernatant. We centrifuge them for another 50,000 revolution for 2 hours, and then you will see the post microsomal supernatant, which is floating on top of the microsomes. Next is the use of the cell-free systems. Liposomes are some of the vesicles whose surface contains an artificial bilayer that is created from purified phospholipids. With these cell-free systems, the buds and vesicles can be produced when purified proteins normally on the cytosolic surface of transport vesicles in the cell are added. It can help us to study proteins. These proteins that bind to the membrane to initiate vesicle formation, those responsible for cargo selection, and those that sever the vesicle from the donor membrane. Using the cell-free systems, we will also see the formation of coated vesicles in a cell-free system. Another type of to study the endomembranes is the use of the mutant phenotypes. So we screen for mutant yeast cells that has exhibiting an abnormal distribution of cytoplasmic membranes, and it reveals proteins that function in secretion. So we will select the proteins that will help these yeast cells to release such molecular compounds out of their system. So we will use the genetic mutants in the study of secretion. So the figure on your letter A is a rough endoplasmic reticulum in the vesicle budding, and it reaches to the Golgi complex where the vesicle has been targeted and fusing with the components coming from the rough endoplasmic reticulum. RNA interference is used to inhibit mRNA translation into proteins. So the RNA interference will block the mRNA translation to form your polypeptides. And by using your siRNA libraries, one can find genes involved in various steps of the secretory pathway. So in this figure, it inhibits the gene expression with the use of the RNA interference, or the RNAi. In essence, your endoplasmic reticulum is a network of membranes that penetrates much of the cytoplasm and has a lumen separated from the cytosol by the ER membrane. It has highly dynamic structures, and it comes with the two compartments that share some proteins and activities. First is the rough ER, which it has ribosome bound to its cytosolic surface. It's flattened, so it's forming a cisternae. It has been connected to neighbors by helicoidal membranes, which is continuous with the outer membrane of the nuclear envelope. So it is parallel to the nuclear membrane. It has a series of connection intertwined to the nuclear envelope. With your smooth endoplasmic reticulum, it doesn't have a ribosome. The membranes are highly curved and tubular, and is continuous also with the rough endoplasmic reticulum. The figures here are showing you the rough endoplasmic reticulum in connection with your smooth endoplasmic reticulum. So look at this figures. This figure will show you the RER is more flattened and the smooth ER is more tubular. Although it doesn't have ribosome, but in structure of these two reticulum, they share the same properties. Starting off with the SER, it includes steroid hormone synthesis in the endocrine cells of your gonad, and especially coming from the adrenal glands or the adrenal cortex. Detoxification of organic compounds in the liver via your oxygenases and enzyme, including your cytochrome P450 family, with the sequestration of your calcium ion and regulated release, showing you this figure of your smooth endoplasmic reticulum. The rough endoplasmic reticulum and the nucleus are near the basal surface, so they are facing the blood supply. The RER is also the starting point of the biosynthetic pathway for secretory protein, since it's being attached to the nucleus. About 1/3 of the proteins are being created at the RER, and released into the ER lumen in a process called the co-translational translocation. Remaining series of proteins and amino acids are synthesized on free ribosomes in the cytosol, showing you the figure of a polarized structure of a secretory cell. So you will see here your mucigen granules, your Golgi complex, your nucleus, and your mitochondrion. Same thing with your rough endoplasmic reticulum. The rough endoplasmic reticulums is creating proteins on membrane bound versus on a free ribosome. So there are two manners where the proteins are being created, one which is in membrane bound, one is on a free ribosome. So the site of this protein synthesis is being determined by the sequence of amino acids in the N-terminal portion of your long polypeptides or series of amino acids. Since secretory proteins contain a signal sequence at the N-terminus, it directs the emerging polypeptide and ribosome to the ER membrane. So these polypeptide moves to the cisternal space of the ER through a protein-lined aqueous channel in the ER membrane as it is being synthesized co-translationally. So there are two ways, okay, to produce this proteins. Proteins contain built-in address codes for protein trafficking pathways throughout the cell. So this has very specific portions or locations where the proteins are being created, plus when they are being shipped to, it's either outside the cell or a part of the cell membrane or part of the cytoplasmic membrane. So this is what they call as your signal hypothesis. So we have here the synthesis of secretory lysosomal or plant vacuolar proteins. Co-translational translocation deposits protein into the lumen of your ER. Polypeptide signal sequence 615 hydrophobic amino acid residues will target the polypeptide to the endoplasmic reticulum membrane. Signal sequence has been recognized by your SRPs. So there are SRPs that are reading the signal sequence. SRPs binds polypeptide and the ribosome and arresting synthesis. So there is also a complex which is recruited to the ER membrane through interactions between the SRP and the SRP receptor on the ER membrane. Once the ribosome is handed off to the translocon, a protein channel is embedded in the ER membrane, and upon attachment, the signal sequence is being recognized as the polypeptide is inserted into your translocon channels. You have here a schematic model of the synthesis of the secretory protein or a lysosomal enzyme on the membrane-bound ribosome of the rough endoplasmic reticulum. So there you see a ribosome which has two portions, the large subunit and the small subunit. This is your mRNA in red, and then the signal sequence on the nascent polypeptide, which is being read through the tRNA, and then the SRP comes along the way. It attaches to the SRP receptor. It forms a plug in the translocon. Then the SRP will pull the ribosomes in between near the ER lumen or the ER membrane. SRP is being released, and the protein plus your ribosomes will be attached with the use of your BIP or on other chaperone to your ER membrane. So this translocon will pull it off. Okay. Several of the steps involved in the synthesis and trafficking of the secretory proteins are being regulated by the binding of or hydrolysis of your GTP. SRP and the SRP receptors are G proteins that interact with one another in their GTP bound states. GTP hydrolysis triggers the release of the signal sequence by the SRPs. Showing you a figure of a schematic model of synthesis of your secretory protein or even a lysosomal enzyme on the membrane-bound ribosome of the rough endoplasmic reticulum. So we have here a pore ring and the plugs and the cytosols. Then the signal protein and signal peptide is being removed from the most nascent polypeptides by signal peptidase, while carbohydrates are being added by your oligosaccharyl transferases. Both of these enzymes Signal peptidase and the oligosaccharyl transferase are integral membrane proteins associated with the translocon. The ER membrane provides a large surface area for the ribosomes to attach, and the lumen gives a specialized local environment that favors protein processing. So this is the synthesis of integral membrane proteins. You know that proteins that are spanning through the membranes on the ER-bound ribosome. So there are integral membrane proteins which are synthesized co-translationally, and their hydrophobic transmembrane segments are shunted from the translocon into the lipid bilayer. So there are, since you will see that it is separation between the cytosol and the membrane and the inner portion of the ER, okay, you will see this figure um, during the membrane protein synthesis. The inner lining of the translocon will orient the nascent polypeptide so that the more positive end faces your cytosol. This is a schematic again, models for the synthesis of an integral membrane protein. In multi-spanning proteins, the sequential membrane segments typically have an opposite orientation. Okay, so you will see that in the 4A. So the arrangement in the membrane is being determined by the direction in which the first segment is being inserted. Tail-anchored proteins lack a signal sequence but are synthesized in the cytoplasm, and they are being targeted to the ER through interactions with proteins in the guided entry of tail-anchored proteins pathway. This is the membrane biosynthesis in the ER. It has been noted that membranes arise from pre-existing membranes. Membranes are enzymatically modified as they move from the ER into another cellular compartments. Membranes are asymmetric with cytosolic phase, and they have also a luminal or extracellular phases established in the ER. So we have to maintain membrane symmetry. We have to make sure that the membrane is asymmetrical. Okay. Membranes of different organelles have different lipid compositions. They can change their lipid and composition by first is using lipid modifying enzymes to convert a phospholipid to another, preferentially including or excluding phospholipid vesicles, exchanging lipids between organellar compartments using the lipid transfer proteins. So we have here modifying the lipid composition of the membranes. In one, there is an enzymatic modification of the lipid head-tail groups. It forms a vesicle, and it can be shunted and transferred to another membrane. Nearly all the proteins are produced on the rough endoplasmic reticulum will become glycoproteins that has attached or added sugars. The addition of sugars to an oligosaccharide is catalyzed by the glycosyl transferases. Each transfers a specific monosaccharide, for example, glucose to the growing end of the carbohydrate chain. The sugar arrangement of the in the oligosaccharide chains of the glycoprotein depends on the spatial localization of enzymes in the assembly line. So these are the steps of glycosylation in the RER. Okay, in the synthesis of the core portion of the N-linked oligosaccharides in the rough ER, you will see here the models, okay, of the steps. We have here the blue triangles, the mannose, the dolicol phosphate, the red diamonds are the glucose, and then the circles are your N-acetylglucosamine or the NAGs. So this is the steps of the synthesis of the core portion of the N-linked oligosaccharides in the rough ER. Soon after the transfer to the polypeptide, the oligosaccharide is gradually modified as the glycoprotein goes through a system of quality control to determine its fitness for specific compartment. Misfolded proteins will be glucose tagged, mannose deficient, and ultimately degraded by your proteasomes. This is what we call as your ER associated degradation. They're making sure and of quality control that the misfolded proteins do not proceed forward. Okay, that is also a part of the clinical case study we've talked about last chapter 2 in your in the biochemistry of life of these proteins that causes the Alzheimer's disease, okay, and the neurofibrillary tangles and dementia. So this accumulation of misfolded protein triggers the unfolded protein response, or what we call as the UPR. Sensors in the ER are kept inactive by the chaperone BIP. When these misfolded proteins will accumulate, the BIP is incapable of inhibiting the sensors. Activated sensors send signals to trigger proteins involved in the destruction of your misfolded proteins. So you will see a model here of the mammalian unfolded protein response, or the UPR, where this misfolded proteins are being acted upon with the translation factor. The translation factor has been phosphorylated, which attaches to the small ribosomal subunit. Then it will inhibit the protein synthesis by your ribosomes. The same thing other way around when it reaches the Golgi complex, where when it reaches the Golgi complex, these proteins are being shipped, okay? But since the sensors are inactive, then it will not be shipped to your Golgi bodies or Golgi complex. So this is the ER to Golgi vesicular transport. The ER to the Golgi complex is the first step in the vesicular transport. RER have specialized exit sites where transport vesicles are formed without the use of ribosome. Transport vesicles fuse with one another and form the endoplasmic reticulum Golgi intermediate compartment going to the Golgi complex. As speaking of the Golgi complex, it's a stack of latent cisternae. They have several functionally distinct compartments. We have the cis phase of the Golgi body faces the ER. The trans phase is on the opposite side of the stack. So this is a figure coming from the book. You will see that the trans Golgi network, the trans cisternae, the medial, the cis, and the cis Golgi network, or the CGN. The cis Golgi network functions to sort proteins for the ER or the next Golgi station. On the other hand, the trans Golgi network functions in sorting proteins to the plasma membrane or to another intracellular destination. So this is more likely to have like another portion of another organelle inside your cell. The Golgi complex is not uniform in composition. There are differences in composition from the cis to the trans phases. So you will see a figure of the regional differences in membrane composition across the Golgi stack. We have also here steps in the glycosylation of a typical mammalian N-linked oligosaccharide in the Golgi complex. So there are assembly of sugars found in the glycolipids and the glycoproteins that takes place in the Golgi. And the sequence of incorporation of sugars into these oligosaccharides is being determined by, again, your glycosyltransferases. So you have here different mannoses, okay, in blue triangles. We have purple triangles of galactoses, half circle fucose, yellow squares sialic acid, and the circles or orange circles are the N-acetylglucosamine. In this vesicular transport model, the cargo is being shuttled from the CGN to the TGN in vesicles. In the cisternal maturation model, each cistern matures as it moves from the cis phase going to the trans phases. So you will see here the dynamics of the transport through the Golgi complex. In the current model, similar to the cisternal maturation model, but with vesicle retrograde transports, your Golgi cisternae serve as an primary anterograde carriers. So we talk about the vesicle transports. There are materials that are carried between compartments using your coated vesicles. Protein coats have two purposes. First is to cause the membrane to curve and form a vesicle, and second, select the components to be carried by the vesicles. In this proposed transport membrane compartments of the biosynthetic secretory pathway, you will see here that in A, The rough endoplasmic reticulum are being uh, the components or the substances, the molecules, for example, your steroid molecules are being carried by your COP1, your COP2, okay? Vesicle transports, it will be carried to the VTC and reaching your Golgi bodies, going out to be secreted outside your cell. Okay, so there are coated buds with the help of these COP2 proteins and the cargo receptors, and the coated buds will cleave off a coated vesicle. Coated vesicles is transporting cargo from the ER going to the Golgi complex. So this is a proposed role of the COP2 coat proteins in generating the curvature of the membrane, assembling the protein coat, and capturing the cargo. So this COP2 coated vesicles bud of specialized domains of the ER, and we call it the ERESS, or the ER exit sites. This begins the biosynthetic pathway. So ER export signals found in the cytosolic tails of the transported proteins. All vesicles have two distinct layers, an outer scaffold and an inner layer of adapter, or what we call as your adapter layer, adapter-like proteins. The structure of the outer scaffolds are very different. The subunits of the clathrin lattice, or the three-legged clathrin complexes, overlap extensively. The COP2 lattice do not overlap. Each vertex of the COP2 coat is formed by four edges rather than the three edges coming from the clathrin and perhaps the COP1 coat. So this COP1 vesicles, as we talked the COP2 earlier, is transporting the escaped proteins back to the ER. So this is the other way around, okay, going back to the ER. Organelle proteins are maintained by the retention of resident molecules excluded from the transport vesicles, and being retrieval of these escaped molecules back to their normal compartment. There are resident proteins of the ER contain an amino acid sequence at the C terminus, serving as the retrieving signal. So we don't want to waste every protein that has been lost along its way. Specific receptors capture the molecules and bring them to the ER in COP1 coated vesicles, and each membrane compartment may have its own retrieval signals. So now we transport escape proteins back to the ER using your COP1 coated or recycling vesicles. Okay, so the ER resident proteins bears the KDEL. The KDL receptors will carry on these proteins. It will be attached going back to the RER. Beyond the Golgi complex, sorting proteins at your trans-Golgi network. Sorting and transport of lysosomal enzymes utilizes the clathrin-coated vesicles. These lysosomal proteins are tagged in the cis-Golgi with phosphorylated mannose residue. So you've seen that inside this small portion here on your left figure. And these tagged lysosomal enzymes are recognized and captured by mannose 6 phosphate receptors. We call them MPRs, which are bound by coat proteins. So the process itself here is showing you endocytosis, okay, which targets lysosomal enzymes going to your lysosomes, or the suicide bags of your cell. We sort the proteins at the trans-Golgi network and sorting and transport of lysosomal enzymes. So clathrin-coated vesicles contain an outer lattice composed of clathrin, an inner shell composed of your GGA adapter proteins, which interacts with the clathrin, your MPR, and their G proteins. You will see that on your on the figure on your right, the square on your right, the zooming in. The G protein RF1 GTP used to initiate membrane curvature and to recruit adapters. So clathrin-coated vesicles are being formed at your trans-Golgi network. When we sort and transport non-lysosomal proteins, secreted proteins which are released by regulated secretion may form selective aggregates that eventually become contained in large, densely packed secretory granules. Mature granules are being stored in the cytoplasm until their contents are being released. Following the stimulation of the cell by a hormone or a nerve impulse. We have also plasma membrane proteins, which may use constitutive secretion or one of the two TGN membrane carrier systems. So there are different in this targeting vesicles to a particular compartment. There are different steps that happen between the vesicle budding and the vesicle fusion, including movement of the vesicle toward a specific target compartment, mediated largely by your microtubule, cytoskeleton, and their associated motor proteins. Tethering vesicles to the target compartment are also mediated by a diverse collection of tethering proteins. Docking vesicles to the target compartment, vesicle and target compartment membranes come into close contact via interaction between integral membranes of the two membranes. There's also fusion between vesicle and target membranes. Going to the particular component, RABs are a family of a small g protein that cycle between an active GTP bound state and an inactive GDP bound state. GTP bound RABs associated with membranes by a lipid anchor. Over 60 different RAB genes are identified in humans. Different RABs become associated with different membrane compartments. So we have here a proposed steps in the targeting of transport vesicles to the transport and the target membranes. So we see here this fibrous tethering complex, the RAB, your GTP, and then attaching to the transport vesicle or the multi-protein tethering complex, your RAB and the GTP, the V snares, T snares, and going to the target membranes. Okay. Also with snares, they constitute a family of proteins localized to specific subcellular compartments. Snares are integral proteins that bring the vesicle and target compartment in close contact. So it pulls the transport vesicle going to the target membrane. V snares are found in transport vesicles and T snares are located in the target compartments. In nerve cells, there are plasma membranes that contains two T snares. We call them the syntaxin and the SNAP25. Synaptic vesicle membrane contains also a single V snare or the synaptobrevin. T and V snares molecules interact to form four-stranded bundles, each with a 4 alpha helices. These parallel alpha helices zip together to form a tightly interwoven complex that pulls the two opposing lipid bilayers into a very close association. So you will see this figure of the models of the interactions between the V and the T snares leading to the membrane fusion and also with exocytosis. Moving forward with exocytosis, it's a discharge of a secretory vesicle or granule after fusion of the plasma membrane. This is triggered by an increase of calcium ions. Contacts between vesicle and plasma membranes lead to the formation of fusion pores. Then the luminal part of the vesicle membrane becomes the outer surface of the plasma membrane, and the cytosolic part becomes the inner surface of the plasma membrane. With engineering linkage, these extracellular vesicles for drug delivery, these extracellular vesicles can move throughout the body unnoticed by the immune system, passing through a lot of difficult biological barriers such as your blood-brain barrier. Their downside is their short half-life before phagocytosis. Currently, these extracellular vesicles can be isolated from cell culture using the ultracentrifugation or affinity purification and loaded with therapeutic RNAs or small other molecules. The challenging now is finding ways to target specific cells in your body. Moving forward with lysosomes, it contains at least 50 different hydrolytic enzymes produced in the RAR and can hydrolyze virtually every type of biological molecule, proteins, nucleic acids, even sugars and lipids. Lysosomal enzymes contain also acid hydrolases that have optimal activity in your acidic lumen. These are the enzymes: phosphatases, including acid phosphatase that has a substrate of phosphomonoesters, acid phosphodiesterase that has phosphodiesters, nucleases such as acid ribonuclease and substrate with RNA, acid deoxyribonucleases with the DNA, cathepsin that has substrate of proteins, collagenases with collagen. You'll have your GAG hydrolyzing enzymes such as your iduronate sulfatase with substrate of dirmatan sulfate, beta galactosidase with keratan sulfate, heparan N sulfatase such as heparan sulfate, the alpha and acetyl glucosaminidase such as with substrate heparan sulfates, follow, polysaccharides and oligosaccharides, alpha glucosidase that has a substrate of glycogen, fucosidase such as fucosyl oligosaccharides, alpha manosidase with manosyl oligosaccharides, sialidase which has a sialy oligosaccharides, ceramidase with ceramides, glucocerebrosidase, glucosylceramide, beta hexosaminidase which has GM2 ganglioside, aryl sulfatase A with galactosyl sulfatide, acid lipase with triacyl glycerols, phospholipase with the phospholipids. Single-celled organisms and their phagocytic white blood cells digest ingested materials by fusing phagosomes and lysosomes. These lysosomes play an integral role in the regulated process of organelle turnover known as autophagy. A phagophore envelope an organelle to produce a double membrane sequestering vesicle called an autophagosome. It fuses with a lysosome generating an autolysosome. Both the inner membrane of the autophagosome and the enclosed contents are being degraded. What are the benefits of autophagy? It helps the organism or protect an organism against intracellular threats such as abnormal protein which aggregates or an invading bacteria. They may also play a role in the prevention of certain types of cancers and even slowing the body's aging process. With green cells or the plant cell vacuoles, this is very special. A vacuole is your membrane-bound fluid-filled compartment. Plant vacuoles have a several storage modification, storage of solutes and macromolecules, storage of toxins, ionic concentration capability, and the vacuole membrane or the tonoplast contains an active transport system to keep high concentration of ions so the water enters by osmosis. Especially in plants living in or thriving in deserts, these plant vacuoles contain hydrolases for degradation of your biomolecules. The endocytic pathway, where moving membrane and materials into the cell's interior. This is the other way around where the opposite of exocytosis. There are two basic processes which has different mechanism. Endocytosis is primarily a process by which the cell internalizes cell surface receptors and is bound to extracellular ligands. Phagocytosis is the uptake of a particular or particulate matter. With these two categories of endocytosis is being discussed, we have bulk phase endocytosis or pinocytosis or the non-specific uptake of extracellular fluid. We have also receptor-mediated endocytosis or the clathrin-mediated endocytosis, which brings about the uptake of specific extracellular macromolecules following binding to receptors on the surface of your plasma membrane. RME and the role of your coated pits. Substances that enter the cell through clathrin-mediated RME become bound to a coated pit on the plasma membrane. We have also a clathrin-coated regions that invaginate into the cytoplasm and then pinch free of the cytoplasm. Each of the clathrin molecules have 3 heavy chains and 3 light chains that join together at the center to form a 3-legged assembly called a triskelion. They overlap and each of the legs of which extends outward along 2 edges of a polygon. Each vertex of a polygon contains a center of one of the component triskelions. Coated vesicles contain adapters between the clathrin lattice and the surface of the vesicle facing the cytosol such as the AP2. AP2 adapters, as shown in this figure, contain multiple subunits having different functions, and they engage in the cytoplasmic tails of specific receptors to select bound cargo molecules, and they bind and recruit the clathrin molecules of the overlying lattice. Coated vesicles may also contain dozen of different accessory proteins that form a dynamic network of interacting molecules. These proteins have roles in cargo recruitment, coat assembly, membrane bending and invagination, interaction with cytoskeletal components, vesicle release, and also membrane uncoating. Dynamin is a G protein required for the fission of the vesicle form from the membrane on which it forms. It self-assembles into a helical collar around the neck of an invaginated cockpit. So you will see here different step of your dynamin subunits forming a clathrin lattice, the dynamic ring, and GTP hydrolysis. So we talk about the role of phosphainositides in the regulation of your coated vesicles. AP2 adapters will normally exist in the cytosol in a locked conformation. It needs the AP2 complex to cause a conformational change. Thus, the AP2 cargo binding site will become exposed and allowing it to interact with specific membrane receptors. Showing you a structural model depicting the charges and the changes in protein conformation that occur upon AP2 binding to the plasma membrane. So there is this AP2 with the Phe52 rich membranes, attraction to membranes, and then the membrane opening and bindings, and thus an open conformation that regulates the cargo binding. After internalization, these vesicle bound materials are being transported in vesicles and the tubules known as your endosomes. Early endosomes are located nearly in the periphery of the cell. It sorts materials and sends the bound ligands to the late endosomes. This is the endocytic pathway, such as you can see here the low density lipoproteins or the LDLs, which are a complex of cholesterol and proteins. The LDL receptors are being transported to the plasma membrane and bound to a coated pit. They are taken up by the RMEs and taken to the lysosomes, which releasing the cholesterol for use by the cells. Lastly, this is moving membrane and materials into the cell's interior. One good example of it is the phagocytosis, which is carried out by the cells specialized for the uptake of relatively large particles. The folds fuse to produce a vacuole, we call it the phagosome. It pinches off inwardly from the plasma membrane and fuses with the lysosome or the phagolysosome. Mammals have a professional phagocytes. We call it the macrophage and the neutrophils. They phagocytize invading organisms, damaged and dead cells. Lastly, this post-translational uptake of proteins by your peroxisomes, the mitochondria, and the chloroplast. These organelles import proteins through one of the more outer boundary membranes. As in the case of the rough ER, those proteins are imported by these organelles that contain amino acid sequences, and they serve as addresses that are being recognized by receptors at the organelle's outer membrane. Unlike RER, which generally imports its proteins co-translationally, the proteins of these other organelles are imported post-translationally, following their complete synthesis on free ribosomes in the cytosols. With uptake into the peroxisomes, they have two subcompartments in which an imported protein can be placed. First one is the boundary membrane and the internal matrix. This possesses a peroxisomal targeting signal. It's either a PTS for a peroxisomal matrix protein or an MPTS for a peroxisomal membrane proteins. PTS receptors bind to the peroxisome destined proteins in the cytosol and shuttle them to the surface of the peroxisome where they can enter the organelle. This outer mitochondrial membrane includes a protein import complex or the TOM complex, which includes a receptor and a channel. Proteins destined for the inner mitochondrial membrane engage with, you'll see here, another protein import complex such as the TIM complex. They are shown in green and yellow. Lastly, uptake into the chloroplast. Outer and inner envelope membrane contains translocation complexes such as the TOC and the TIC to facilitate import of the proteins. We have also seen here in the figures chaperone that unfolds the protein in cytosol and fold them in the chloroplast. Proteins also include a transit peptide sequence. That's all we have for today for the binding boundaries, the structure, and the function of their cytoplasmic membrane. This is your Cell and Molecular Biology lecture series. Have a nice day, everyone. Thank you.